viewers, today I will talk about enzyme kinetics, regulation as well as enzyme catalysis. I will talk about the active site of the enzyme, coenzyme, nomenclature, catalysis, kinetics, inhibition, regulation as well as special classes of enzyme. So, enzymes are biocatalysts regulating the homeostasis of the cell. Within the cell, it regulates the metabolism. Outside to the cell helps in blood clotting, and plasma membrane enzymes are responding towards the extracellular signal. So, active site is the three-dimensional crevice or pocket of the enzyme which fits to the substrate, which has some configurational change and stabilizes the transition state. Now, it has two models. Number one, Fischer lock and key model, and number two, Koshland's induced fit model. In 1898, Emil Fischer had proposed the lock and key model where already pre-shaped template is present. Okay, so perfectly complementary substrate to enzyme. But in 1958, Koshland had proposed induced fit model where the contact of enzyme induces the configuration and conformational changes within the enzyme active site to fit properly to substrate and to carry on the catalysis. Coenzymes and cofactors. Cofactors are non-protein small molecular weight compounds or ion whatever. It actually helps in catalysis. So, they are co-catalysts. It may act as bridging molecule, it can stabilize enzyme substrate complex, or it can help in case of charge transfer as well. It may be organic or inorganic. Organic may be coenzyme or prosthetic group. If it is tightly bound, it is called as prosthetic group, like thiamine pyrophosphate in case of pyruvate decarboxylase. But if it is loosely bound, it is called as coenzyme, like thiamine pyrophosphate in pyruvate dehydrogenase. Inorganic include metal ion as well as iron sulfur cluster. In case of carbonic anhydrase, zinc ion is present. Mg2 plus is present in case of hexokinase as well as it acts as cofactor in case of phosphatase. Iron sulfur cluster is needed for redox signaling. Enzyme nomenclature. So, according to different class, it actually ranges from 1 to 6. Number 1, oxidoreductase. It helps in oxidation reduction reaction. Actually, they are dehydrogenases. Electron can be donated or can be accepted. So, according to donor or acceptor, subclasses are made. 1.1 is alcohol dehydrogenase, where CHOH alcohol is the donor group. Number 2, transferase helps to transfer some specific group, maybe the methyl group 1 carbon group, that helps in case of methyl transferase, that is 2.1 subclass. Hydrolase, that helps in break the hydrolysis of the bond COCN etc. Like in case of dehydratase as well, esterase 3.1. Lyase actually break the bond COCN etc. So, there are so many lyases are there, like dehydratases in case of 4.1 subclass. Isomerases that actually catalyzes the configurational or stereoisomer changes, and in case of 5.1 it is racemase that actually helps in racemization. Ligase, with the help of ATP, it helps to make the bond in between COCN etc. In case of ATP synthase, it is needed to join the bond, phosphate bond. There is a difference in between synthase and synthetase. Synthetase is ligase, but synthase is lyase, needs the help of ATP to join the bonds. Enzyme kinetics, equation, significance and plots. Mechanism of action. So, enzymes actually lower down the activation energy of the reaction. It reduces the entropy, it gives the alternative pathway and reduces the energy for transition state. It is getting stabilized. According to the transition state stabilization, delta G free energy is getting lowered, stabilized by charge distribution, and the dynamics of the protein is actually the protein network at the protein domain as well as active site amino acid. The vibration of them actually is getting concerned related to the allostericity. Enzyme versus substrate plot, okay, substrate concentration. It is more or less saturation kinetics proposed by Brigg and Holden and Henry. What is that? So, initially when the substrate concentration is low, V0, the initial rate of the reaction, maintains the first order kinetics. So, it is linear. But after that, when the substrate concentration is getting increased, then it becomes the mixed order kinetics. Then when the active sites, all the active sites of the enzymes are getting saturated with the substrate, with the high substrate concentration, asymptotically it reaches towards the plateau. So it is the zero order kinetics. Totally the saturation kinetics gives the hyperbolic curve. Now, Michaelis and Menton in 1913 had proposed the equation for enzyme kinetics, where enzyme E plus substrate S gives rise to ES complex by the reversible reaction, ultimately by rate determining step giving rise to free enzyme and product formation. K plus 1, K minus 1, K plus 2, they are all different rate constant. K minus 2 is very negligible. The significance of Km. As we know that Michaelis-Menten equation actually designates the saturation kinetics as well as steady state kinetics. From here, we can observe the Michaelis constant Km. What is Km? Km is the concentration of substrate when V0 or initial velocity is half of maximum velocity. In the hyperbolic curve, ultimately it reaches to the maximum velocity to the saturation level. As well as Km is equal to Ks, substrate affinity, that is inversely proportional, because Km is equal to K minus 1 plus K plus 2 divided by K plus 1. Now K plus 2 is very less than that of the K minus 1. So that is equal to K minus 1 divided by K plus 1. The K is substrate affinity constant, concentration of E into concentration of S divided by concentration of ES. It is inversely proportional. Km is the molar concentration where V0 is equal to Vmax by 2. It is getting increased in case of competitive inhibitor. In case of different isozymes, this is different, but it is indifferent or independent with respect to the enzyme concentration. Other parameters are there because K plus 2 is taken as the K-cat catalysis because it is the rate determining step, the final step to produce the product, as well as K-cat divided by Km is equal to K efficiency, the efficiency of the catalysis. Assumptions, but for Michaelis-Menten there is so much assumptions are there. Product production is linear, no cooperativity, no allostericity is present, and a single enzyme is actually giving rise to the product, and without enzyme no product formation is actually happened. Now, these hyperbolic curve give rise to different scattered non-precision points in higher substrate and lower substrate concentration. For these, the linear conversion to the Michaelis-Menten equation is giving rise by the Lineweaver-Bach plot. That is 1 by V0 is equal to Km by Vm into 1 by concentration of S plus 1 by Vm. So, from this plot, we can observe the slope is Km by Vm and intercept on the y-axis is 1 by Vmax. They can be, Km Vmax can be experimentally derived. Apart from which, the Eddie-Hofstee plot is there where V0 versus V0 by concentration of S can be plotted. In case of Hens-Wolf plot, it is plotted concentration of S by V versus concentration of S. So, they are actually giving different types of intercepts in positive or negative level. Enzyme inhibition. So, with the help of some molecule called inhibitor, enzyme reaction rate is getting reduced. So, they are called as inhibitor. Enzyme inhibition may be irreversible or reversible. Reversible may be competitive, uncompetitive or non-competitive. The first one, irreversible inhibition. Here actually it is making, the inhibitor is making covalent bond with the active site amino acid like serine, cysteine, etc. But it cannot be reversed anyhow. Diisopropyl phosphofluoridate as well as mustard gas, nitrogen mustard, sulfur mustard, they are making adduct with the serine hydroxyl group at the active site by making covalent bond. It is permanently damaged, cannot be reversed back. But reversible inhibitors can be eliminated by dialysis. So, they are making some non-covalent hydrogen bond, polar interaction and so on. So, that can be removed. There are three types that I have already told you: competitive, uncompetitive, non-competitive. In case of competitive reversible inhibition, the inhibitor is structural analog. Okay, so it fits to the active site. So we can find in case of succinate dehydrogenase, where succinate is converted to fumarate, malonate is the structural analog to the succinate. So it acts as competitive inhibitor. So Km is increased as well as Vmax will be unaltered because E plus I is equal to EI. The inhibitor is only bound to the free enzyme. Uncompetitive inhibition. Here actually the inhibitor is bound to the enzyme substrate complex. That may be possible by different drugs also. So in case it actually lowers down the Km, but Vmax will also be reduced here. So graph is actually telling us about the uncompetitive inhibition. From the graph also, the non-competitive inhibition says that it is bound not only to the free enzyme, okay, because in case of uncompetitive, ES plus I giving rise to ESI. Here in case of non-competitive inhibitor, it binds to both free enzyme as E plus I is equal to EI as well as ES plus I is equal to ESI. That actually unalters the Km but decreases the Vm value. This is actually found in case of EDTA, which binds to the magnesium or Mn plus 2 ion, which is responsible as the cofactor in DNase as well as many other enzymes. Next one, enzyme regulation, allosteric control, covalent modification as well as protein processing or covalent activation of zymogen. Number 1, allosteric control. Allosteric modulator a non-substrate compound binds to the allo site. Allo mean other than catalytic site but regulates the catalytic activity at the active site. In case of aspartate transcarbamylase, which actually catalyzes the first step in the CTP biosynthesis or purine pyrimidine biosynthesis. From carbamoyl phosphate plus aspartate, it is giving rise to carbamoyl aspartate. Here we can find it is a kind of allosteric enzyme because it has 3 regulatory subunits which are dimeric and 2 catalytic subunits which are trimeric. Okay, so the CTP final product, it actually binds to the regulatory site and can decrease the activity, as well as ATP is the heterotropic effector that increase the activity by binding to the regulatory subunit as well. And we know that according to kinetics, it does not follow the Michaelis-Menten equation, okay? It follows the cooperativity, that is binding to one subunit increases or decreases the active site activity. Positive cooperativity is found in case of ATP binding, but negative cooperativity is found in case of CTP binding. T state is actually giving the lower substrate affinity, but R state giving rise to the higher substrate affinity over here. Feedback inhibition. In multi-step metabolic pathway, what is getting happen? The final product, they can inhibit the first initial step enzyme, the rate determining step enzyme. Here, from the threonine, the conversion to the isoleucine is a multi-step process, but the isoleucine itself can inhibit the first enzyme threonine deaminase or dehydratase, which actually helps to convert threonine to alpha ketobutyrate. So this is called feedback inhibition. Covalent modification of the enzyme. So the active site amino acids are there. So covalently they can be modified by the help of kinase by addition of phosphate group, or demodifying enzymes are there. They are called as phosphatase, which can remove phosphate group. In case of glycogen phosphorylase, there is presence of serine residues having hydroxyl group. So phosphorylase B is in active form. So with the help of kinase, it can be converted to phosphorylase A having phosphorylated group. So where all those phosphate group can be removed once again by phosphatase according to the signal cascade. So in this way, glycogen phosphorylase can control glycogen synthesis or glycogen breakdown as well by covalent modification of all those kinase and phosphatases. Protein processing or zymogen activation by covalent modulation. So chymotrypsinogen and so many other zymogens are there. When they are getting synthesized, they are as zymogens, but in different pH environment, some of the amino acids are hydrolyzed, they are removed as peptides, and then only the active site can be revealed. So from chymotrypsinogen, pi chymotrypsin as well as alpha chymotrypsin is getting formed, giving rise to the active form of chymotrypsin. Enzyme catalysis, bond strain. So it is actually the preferential state binding which helps to stabilize the configurational ES, transition state complex. Bond strain means a structural rearrangement to the substrate which mimics to the transition state complex. So here we can find the chair boat structure of the sugars can be made during the course of catalysis. Proximity or orientation effect. So intramolecular reaction is actually facilitated. Here you can find in case of paranitrophenyl phosphate reaction. Intramolecular reaction is facilitated than that of the intermolecular reaction. So that is called the ankymeric assistance, apart from which we can find the precise orientation or alignment of enzyme catalytic group as well as substrate, which is called as orbital steering. Electrostatic catalysis. So, by formation of the ionic bonds which actually help to stabilize the transition state configuration. That may be possible via acid-base catalysis or metal ion catalysis. Acid-base catalysis, once again the ionic form is stabilized the transition state. That may be special or specific or maybe general acid-base catalysis. General acid-base catalysis is found in case of RNase catalysis. There are two very consensus conserved histidine residues are there, histidine 12 and histidine 119. Having imidazole group, they can act as general acid as well as general base, which facilitates the RNA hydrolysis as well. Metal ion catalysis. So, in case of carbonic anhydrase, we can find the presence of zinc ion, which helps in the nucleophilic attack of carbon dioxide, which is actually stabilized by 3 imidazole ring contributed by the histidine residues of the enzyme itself. Covalent catalysis. So, covalent bond is made which stabilizes the transition state. Serine hydroxyl classes a silyl enzyme as chymotrypsin. Phosphoenzyme. In case of cysteine, thiol group is contributed to make the covalent bond. In case of glyceraldehyde 3-phosphate dehydrogenase. In case of histidine, imidazole group is actually contributed in case of succinyl CoA synthase to make the transition state complex. In case of lysine, extra NH2 is getting involved in case of fructose diphosphate aldolase. Next one, quantum tunneling. So, in all the cases, actually, we find the over the barrier reaction is facilitated by enzyme catalysis process. But quantum tunneling is through the barrier process. Ribozyme. As we know that all enzymes are protein in nature, but the ribozymes are RNA, which are catalytic in nature. So, they are called as ribozyme. Ribosome itself is a huge machinery containing ribozyme as peptidyl transferase. And RNase P helps in case of splicing, editing. It is also important. Ribozyme. Abzyme, the catalytic antibodies, okay, that means CatMab, monoclonal antibody. They help to bind with the substrate specifically or antigens, and they help to form transition state stabilization and product formation. It is very much important in HIV treatment as well. Isozyme and allozyme. So, isozymes are different forms of enzyme. They differ according to different kinetic parameters as Km can be changed. That is found in case of lactate dehydrogenase. So many different isozymes are there, starting from skeletal muscle M4 to the heart muscle, that is H4. The in-between isozymes are M3H, MH3, M2H2, etc. Allozyme, they are getting synthesized from different allele, from different loci. But bisubstrate reaction means two substrates are reacting with the enzymes. So, it may be single displacement or double displacement. In case of single displacement bisubstrate reaction, that may be sequential, one by one. So, that may be ordered sequential or random sequential. In case of double displacement reaction, it follows ping-pong reaction, okay, ping-pong bye-bye reaction. One particular substrate donates some group, leaves as product, the next substrate comes and takes out that particular thing, and then it is getting converted. That is actually happen in case of transaminase reaction by the presence of pyridoxal phosphate, which is getting converted to pyridoxamine phosphate. So, student, I have talked about the different types of enzyme nomenclature, catalysis, as well as specific points regarding enzyme kinetics. Thank you.